welcome to another session of CEC Gurukul lecture series. Today we shall be talking about the ovule in angiosperms. As you are already aware that sexual reproduction in angiosperms takes place by fusion of two gametes, male gamete and a female gamete. Male gamete we have already talked about in earlier lectures, and this male gamete is present in the pollen grain, which is the male gametophyte. The female gamete is the egg, and this is present in an embryo sac which is housed in the ovule. So, ovule is basically a sporophytic structure which is attached to the placenta, and it is attached to the placenta by a small stalk, and it is the funiculus. Now, the ovule consists of a nucellus tissue which occupies the major portion of the ovule, and this nucellus is protected on the outside by two coats which are called as the integuments. The nucellus here is equivalent to a megasporangium, as it encloses a haploid megagametophyte, the embryo sac. The embryo sac harbors the female gamete or the egg. So, the main function of the ovule is to produce a seed. Depending on the number of ovules, the number of seeds vary in angiosperms, and each seed after germination will develop into a seedling, because seed is the one which contains an embryo. Most ovules also contain an endosperm. Endosperm is basically a tissue which is required for nutrition of the developing embryo. Ovule is a multicellular oval structure which originates from placenta, like I just told you a few minutes back. It lies in the chamber of the ovary, and this ovary can contain more than one ovules. The number of the ovules varies in different species. The mature ovule, like we just discussed, consists of a body and a stalk, which is the funicle. Now, the point of attachment of this funicle with the ovule body is what is called as the hilum. Funiculus sometimes extends up to the base of the ovule, and the ridge which is hence formed is called as a raphe. Now, you can see the structure of an ovule in this particular diagram. The ovule is divided into three regions. It's the distal, median and the proximal region. The distal region basically occupies or contains the nucellus along with the two integuments. The median portion is the chalaza, which is the point of attachment. Also, it's the region where the vascular bundle or the vasculature of the ovule enters the ovule, and then there is the proximal region which contains the funiculus along with the vascular supply. Two integuments, they are totally enclosing the nucellus, but there is some point where the integuments are not actually enclosing fully the ovule, and this is a region which is left open, or it is a pathway which is created, and this is called as the micropyle here in the ovule. So, though the two integuments completely surround the nucellar tissue, they leave a narrow opening at the apical end, and this narrow opening is called as the micropyle. Pollen tubes reach the nucellus by entering through the micropyle, and then eventually they enter the embryo sac through one of the synergids. There are certain species where there is no micropyle which is formed, so but those are exceptions. The nucellus is important because the nucellus harbors the meiocyte, which will eventually undergo meiosis to form the megaspores. The portion of the micropyle is contributed by both the integuments, while the portion contributed by the outer integument is called the exostome. The one which is formed by the inner integument is the endostome. One more thing which I would like to mention here is that micropyle is a narrow passage which is meant for the entry of the pollen tube into the ovule, and it is aligned. In Ricidaceae family, the micropyle is in a zigzag manner. Now, based on the curvature of the ovules, that is the position of the micropyle and the chalaza, there are different types of ovules. In this particular diagram, you can see many different types of ovules, starting with orthotropous ovule on the left hand side, where you see that the micropyle and the funiculus, micropyle, chalaza, they are in one straight line. In the next one, you can see that the funiculus has elongated and it has brought down the micropyle closer to the funiculus. In the rest, there is a typical curvature. Now, depending on the degree of the curvature, the ovules are named differently. So, we will take these up one by one. Orthotropous ovule is also called as the atropous ovule. Ortho means straight. So, the body of the ovule here is erect. The hilum, the chalaza, the funiculus and the micropyle lie in a straight line. Hilum is basically a scar which is left where the funiculus is attached. Okay. So, polygonum is one example of orthotropous or atropous ovules. Anatropous ovule is the one where the body of the ovule, it becomes completely inverted during the development. So, what happens because of this curvature is that the micropyle lies very close to the hilum or to the funiculus, and this is the most common type of ovule which is found in angiosperms, because it occurs in nearly 82% of the angiospermic families. Then comes the hemianatropous or hemitropous ovule. Now, the body of the ovule is placed transversely, or we can also say that it is present at right angles to the funicle. The micropyle and chalaza, they lie in one straight line. This is seen in ranunculus. Now, if the body of ovule is curved or even more bent than what we observed, so the micropyle and chalaza, they do not lie in the straight line, then such ovules are called as the campylotropous ovules. In campylotropous ovules, the curvature is less than what we observed in anatropous ovules, and such ovules are seen in Leguminosae family. Then there are amphitropous ovules. Now, you have to remember that amphitropous ovules are very important, and here the curvature of the ovule is also seen in the embryo sac. That is, the curvature of ovule is pronounced in the embryo sac also, because here the embryo sac also becomes curved, and many a time this embryo sac looks horseshoe shaped. This is seen in family Butamaceae. Circenotropous ovule. Now, this circenotropous word has come from the word circle. The nucellus and the axis are in the same line in the beginning as we observed initially, but due to rapid growth on one side, that is, the funiculus is growing laterally on one side and the curvature continues so as to the extent that it goes around 360 degrees and the micropyle again points upwards. So, these ovules are called as the circenotropous ovules, and these are seen in Opuntia. Okay. Now, I would just like to go back and show you some of these drawings here. So, you see here on the right hand corner there is this circenotropous ovule. So, the first one is orthotropous, then anatropous, hemianatropous, circenotropous, and down below you can see the curvature is even pronounced in the nucellus. Okay. So, these are the different types of ovules which are based on the curvature. Now, coming to the components of ovules, Like we have just discussed, that ovule consists of integuments, mostly two. Then there is endothelium, micropyle, nucellus, funiculus, obturator, and hypostase and epistase. As we know that when we study the general structure of an ovule in angiosperms, it has two integuments, that is, it has two layers of covering. Most angiosperm ovules, they have two integuments, and such ovules where there are two integuments present, these are called as the bitegmic ovules, seen in monocots, and we can say that polypetally members are also examples of bitegmic ovules. If there is only one integument which is present, then we say that the ovules are unitegmic. Now this presence of only one integument can be either because both the integuments they fuse together, as is seen in Myrtaceae, or it can be due to suppression of any one of the integuments, seen in Ceratophyllaceae, Asteraceae, and many other families. The third category is where the ovules, they do not have any integuments as all, as in Oleaceae. So such ovules which lack integuments are called as the ategmic ovules. Another important thing which I would like to mention here about the integuments is that in some families, like in Cactaceae, there are some air spaces which are present between the two integuments. That is, there is a prominent air space which is present between the outer and the inner integument. In Cleome and Magnolia, the integuments, they have stomata present on them and they can perform the function of photosynthesis. Chlorophyll is also present in Hymenocallis and Gladiolus integuments. You can see here in the first one there are two integuments, hence the ovules are called bitegmic. Then we have just one integument, so which represents a unitegmic ovule, and third one is again a unitegmic ovule, but the tip of the integument is divided into two lips, or it is called as the bifid integuments. Then inner to the integuments we talk about the endothelium. This is a specialized tissue which is present around the embryo sac, and it is important because it supplies nutrition to the embryo sac, and this nutrition comes once the nucellus has degenerated. So endothelium organizes from the innermost layer of the inner integument, right? So it's basically a part of the integuments only. Since it is being formed by the integuments, it is called as the integumentary tapetum, and because it is important in supplying nourishment, it is functionally similar to anther tapetum. That's why it is called as the integumentary tapetum. Mostly the endothelium is single layered, but in case of Helianthus, it is multi-layered and the layers can go up to 10 or 12 in number. Cells of the endothelium are radially elongated and they are rich in cytoplasm, and they store both starch as well as fats. Often the endothelium cells are polyploid and they can be up to 32N in number. Sometimes they are even multinucleate. The cells of endothelium, they exhibit features of both meristematic as well as secretory cells, because they are performing the function of a nutritive layer. They are high in proteins, RNA, carbohydrates, ascorbic acid, and also in some enzymes. Endothelium is often surrounded by cuticle, and the cells, they show wall thickenings. These wall thickenings are mainly of callose, which separates the embryo sac from the sporogenous tissue. An endothelium like tissue which is instrumental in providing nourishment is formed by persistent nucellar cells in Begoniaceae. and also in Fabaceae. Now these cells of these cells which are providing nutrition, which are arising from the nucellar cells, these are called as false endothelium, because their origin is not from integuments and their origin is from the nucellar cells. Here you can see that the endothelium is present around the embryo sac. The cells have a prominent and pronounced nuclei which are present. This is seen in Brassica species. Then we come to the micropyle. As I just mentioned, the opening at the apex of the integuments is the micropyle, which allows the pollen tube to enter the ovule, which is very necessary for fertilization to occur. The micropyle is divided into two portions. The outer one is the exosome, the inner one is the endostome. In Ricidaceae, the endostome and the exosome are not in a straight line and they give rise to a zigzag micropyle. Sometimes the micropyle can get filled with an exudate, as is seen in Ornithogalum, and this exudate is covered on the outside by a hymen. The exudate is formed by the cells of the nucellus as well as the cells of the inner integument. This exudate attracts the pollen tube to enter the micropyle. So we can say that the exudate is a chemotropic substance which is guiding the path of the pollen tube into the micropyle. Here you can see that there are especially in figure B, C and D, there is an exudate which is present, and this exudate it attracts the pollen tubes into the ovule. Now this is here you see that there is a micropyle which is an opening, then the major part of the ovule is occupied by the nucellus. Nucellus is a multicellular tissue which is present, and the megasporocyte or the embryo sac eventually develops from the cells of the nucellus. Nucellus is the wall of the megasporangium. In most angiospermic families, there is only one nucellus present per ovule, but in eagle marmelos, which is an exception, there are two nucelli which are present. Now, any one particular cell of this nucellus can behave as an archisporial cell, and it develops immediately below the nucellar epidermis. So, this archisporial cell, it remains within the confines of inner integument. Sometimes it can project into micropyle as a small beak, as is seen in Euphorbiaceae and Cucurbitaceae, and the cells of this nucellar beak, they store starch and protein. Nucellar epidermis is forming a cap in the micropylar region of Ornithogalum. The cells are columnar, they are lignified and densely cytoplasmic with high amounts of endoplasmic reticulum and RNA. These cells are mostly consumed by the developing embryo sac or by the endosperm. In certain species, this nucellus is a persistent tissue. It may persist in mature seed as a nutritive tissue, as we can see in black pepper also, and such persistent nucellus is called as the perisperm. It can break down precociously, that is before time, leaving a large cavity around the embryo sac, and this large cavity is called as the pseudoembryo sac, which is a very characteristic feature of family Podostomaceae, and this pseudoembryo sac is the one which nourishes the developing embryo in the absence of an endosperm. Now there are ovule types which are demarcated on the basis of the nucellus. That means that there are two types of ovules depending on the thickness of the nucellus. Nucellus thickness is the number of cell layers above and around the myocyte or the megaspore mother cell, which is used to classify the ovules. So, we have tenuinucellate and crassinucellate ovules. There is another category, third category, which is the pseudo-crassinucellate ovules. So, I will now be explaining you what tenuinucellate and crassinucellate ovules are. In tenuinucellate ovules, the archisporial cell, which is present just below the epidermis of the nucellus, it can directly function as the megaspore mother cell. So, as it is present just below the epidermis, the sporogenous tissue is hypodermal. The nucellar tissue covering it on the outside is just single layered. So, here this is characterized by presence of a single integument, that is unitegmic, and these ovules are more advanced in nature. They are seen in Ericaceae, Lamiaceae, Solanaceae. So, the nucellar tissue here is covering the archisporial cell on the outside as a single layer. The archisporial cell is hypodermal in its position. So, this will eventually behave as the spore mother cell and give rise to the megaspore. The second one is the crassinucellate ovules, where you can see on the right hand side of the picture that the archisporial cell is embedded deeper into the nucellus, that is, it is not hypodermal in origin. There is nucellar epidermis and there are a few more cell layers on the outside of this archisporial cell. So, basic type of ovule in which the archisporial cell divides transversely, those are the crassinucellate ovules. So, when this divides transversely the archisporial cell, it is divided into two cells. There is a parietal cell on the outside and there is a sporogenous cell on the inside. So, if there are further divisions which are happening in the parietal cell, the sporogenous cell is now embedded in the nucellus, and instead of being hypodermal as it was in tenuinucellate ovules, now it becomes subhypodermal. The nucellus is more than one cell layer thick around the myocyte or the megaspore mother cell. There are two integuments present and hence it is bitegmic. Such ovules are seen in Euphorbiaceae and Gingiveraceae. Third category, like I just mentioned, is of pseudo-crassinucellate ovules. Now, what happens here is that here too the sporogenous cell is embedded as in the crassinucellate ovules, but this embedding of the sporogenous cell is not because of division in the archisporial cell, which eventually behaves as the megaspore mother cell. This embedding is because of divisions which are happening in the epidermis. The epidermal cell, it divides by transverse divisions, and hence the archisporial cell becomes embedded because there is an additional layer added because of transverse divisions in the epidermal cell. In figure A, we see the tenuinucellate ovules in Zelenadium, and in B, there are crassinucellate ovules as seen in Hippophae. Another structure which is associated with the ovule is the obturator. Obturator is a very important ovular structure because this directs the pollen tube towards the micropyle. It can vary in its origin. It can arise from the placenta, it can arise from the funicle, and there is a difference in the morphology and the extent of this obturator. Funicular obturator is formed by the local swelling of the funiculus, where cells of funicular epidermis, they elongate, they become densely cytoplasmic as seen in ceratocephalus, and there are glandular epidermal hair which are present on both sides of the funiculus. Now, these epidermal hair which are seen in Tetragonia tetragonoides, they guide the pollen tube into the micropyle, and sometimes the cells at the base of the funiculus, they become secretory. In Drimys and Exospermum, they are secretory, and their secretions guide the pollen tube into the micropyle. There is a knee-shaped funiculus which is present in Crinum. Here you can see in Tetragonia tetragonoides, obturator cells which are present on both sides of the funiculus. They show multicellular and glandular here. Okay. So, we will stop right now here and continue with the rest of the structures in the next lesson. Thank you very much.